All right. Hello, everyone. My name is Anne Flesch, and thank you for allowing us to present our result. I apologize. I am not able to be with you in person to present you this. And thank you to Monsieur Dubernay for the introduction. So today's agenda, of course, will start with an introduction and end with a conclusion, but the core of this presentation will be two experimentation. Experimentation A is the study of the impact of Saccharomyces cerevisiae inoculation timing, and experimentation B will be the study of the impact of Saccharomyces cerevisiae preparation mode. So Saccharomyces cerevisiae starters are widely used in industrial winemaking, and they guarantee the stability and the reproducibility of wine processes. We know through different studies that the choice of the type of yeast, the inoculation time, but also the preparation mode can affect all the yeast behaviors, therefore their kinetics, their population, and have a significant impact on the final wine. So the main experimentation goals of this study were the following. For the impact on the yeast inoculation timing, the main goal was to see how it affected the microbial populations of Saccharomyces and non-Saccharomyces yeast, and eventually, of course, the resulting wine and its aromatic profile. For the study B, the main goals were to see how the preparation of the yeast would impact the implantation of this yeast, but of course also how it affects the resulting wine and its aromatic profile. So let's start with experimentation A, the impact of Saccharomyces cerevisiae inoculation timing. The main protocol is, it was done in the vintage 2020 in Languedoc-Roussillon, France. It was done on Macabeo grape, and the yeast that was chosen for this experiment was Safeno HDA54. That comes from the hybridization program of Le Saf and Fermentis. This yeast specifically was chosen because it has a long lag phase, and it's quite important as a characteristic when you choose to inoculate in pre-fermentation phase, but also another characteristic of this strain that it overexpresses acetate esters, and this will be important when we analyze the resulting wines. Something else important to notice is that the pre-fermentation temperature was maintained below 10 Celsius at all time in order to prevent a start of the fermentation. The fermentation temperature was between 17 and 18 Celsius, and eventually we used pneumatic pressing, and we did sulfiting at 3 grams per hectoliter after pressing. So in this experimentation, we had 4 different inoculation times. The first one was directly on the grape. The first one was directly on the grape at reception. The second one was in the juice after crushing. The third one was in the juice directly after the press. And the scenario number 4 was divided. The 20 grams per hectoliter of yeast were divided equally between after crushing and after settling. In these 4 conditions, the yeast was direct pitch. Only in condition number 1, the yeast was directly just quickly suspended in water before being directly put on the grape. So this is the average must analysis. We have the sugar in gram per liter at 225 gram per liter. And so what's interesting here to notice is the initial YAN was at 96.5 ppm that we adjusted to 175 ppm through the addition of DAP at 1/3 of the fermentation. And we also did at that time aeration. We also adjusted the turbidity of the must. So this is the first result, and it is a microbial population during the pre-fermentation phase. So what you can see here is for each scenario, the analysis of the yeast population at 3 different times, directly after crushing, after pressing, and after settling. And we analyzed 3 different things, so total yeast in bright red, the Saccharomyces cerevisiae yeast population in yellow, and in dark red, brown, we have the non-Saccharomyces yeast population. So we see here also at what time we added the yeast. So what we observe here is that when we add a 20 gram per hectoliter, the HTA54 Saccharomyces cerevisiae yeast, we see with time a decline of all strain, but specifically the non-Saccharomyces cerevisiae strains. And so we can see that it inhibits, but it doesn't kill the non-Saccharomyces. Here we have a focus specifically after settling. This was done a little bit later than in the previous slide, of the population in the 4 scenario. So we can see that the scenario that had the highest yeast population and Saccharomyces cerevisiae yeast population was after pressing, which we assume is the scenario where, because you start being in a true liquid phase at this time, the yeast can develop very quickly. If we look here on this graph, we see the different percentage of the yeast population in the 4 scenario. So we can see that in the scenario addition directly to the grape and post-crushing, we have, out of the total yeast population, approximately 70% is Saccharomyces cerevisiae, and approximately 25–30% is non-Saccharomyces cerevisiae. But out of the cerevisiae population, 100% is HTA54. So by inoculating HTA54 very early on, it allowed to really overpower all the other Saccharomyces cerevisiae yeast population. On the 2 other scenarios, pressing and crushing, we see that we have a higher percentage of Saccharomyces cerevisiae yeast population over the whole population, so between 90 and 100. But HTA54 is still very well implanted. So we have 80 to 90%, but we also observed 2 different strains in this scenario. Look at the kinetics. So the kinetics of the 4 different scenarios, we see that overall they have very similar kinetics, and they all end fermentation between 17 and 19–20 days. The scenario pressing that had the highest population that we observed in the previous slide, you can see that we observe earlier start of the fermentation, a shorter lag phase, and eventually it's a scenario that finished the fastest, which correlates with the previous slide. We analyzed the yeast population at 2/3 of the fermentations, or approximately here where the blue arrow is. And we can see that in the 4 different scenarios, more than 90% of the total yeast population was Saccharomyces cerevisiae, and more than 90% of the total yeast population was HTA54. So this is a very good implantation rate in all scenarios whenever the yeast was inoculated for these 4 fermentations. Here we have Saccharomyces cerevisiae yeast population monitoring during alcoholic fermentation by flow cytometry through the method that was just explained by Monsieur Dubernay. And you can see here that we had 4 different samples during fermentation at day 1, 9, 14, and 17. And so what you observe, first of all, is that we see pretty traditional curves for fermentation, alcoholic fermentation, an exponential phase, a stationary phase, and a declining phase. And something else that we can see is that the scenario pressing also here shows a very high population early on fermentation. This is the analytical parameters of the 4 resulting wine post-fermentation. And we're not going to go into the details, but we can see no significant difference between the 4 fermentation when it comes to ABV, acidity, SO2, pH, and so on. We then analyze the aromatic compounds, and here more specifically the ethyl esters that are responsible for complex fruitiness. We have 4 different compounds that are expressed in other active value. So other active value is their concentration divided by their perception threshold. And what you can see here is that whatever the scenario, we can see no significant difference between the production of these esters whenever the time the yeast was added. Now, the dosage of the acetate esters, remember it's a key signature compound of this strain. We can again see differences, but no significant difference between the four scenarios for the acetate esters. Acetate esters, and more specifically the isoamyl acetate. The next step for us was to do a triangular tasting. So we had a panel of 6 professional tasters, and we compared different scenarios with each other. Scenario 1 versus on the grape versus scenario 3 after pressing, and scenario 1 versus scenario 4, for example. And here you have the result of how many tasters could identify a difference. So because it's a quite small panel, statistically, these results are non-significant. However, we could see that 4 out of 6 tasters could taste the difference between number 1 and number 4. Therefore, we decided to move forward with a descriptive tasting of the wines. So we did a descriptive tasting with 18 different variables, 6 professional tasters, and this was done through ANOVA, analyzed through ANOVA statistic analysis, so analysis of variance. Here we display the result for amylic perception in the mouth, and you can see that all the 4 scenarios are part of the same groups, A and B, and so therefore this means there is no significant difference for these 4 scenarios for this criteria, which is again the key identifier marker for this strain. So eventually, there was no significant difference in the sensory. The main conclusion is that an earlier timing of inoculation of Saccharomyces cerevisiae, and more specifically in this case, HDA54, can allow for a good occupation of the ecological niche without compromising on fermentation performance, analytical profile, and organic profile. So really here, we could see no significant difference on the implantation rate. They were very good in all cases. The fermentation kinetics, the base analytic parameters of the wines, and the aromatic analysis. And when it comes to organic profile, no significant difference. So it does give the winemakers flexibility on when they choose to add the yeast, and in order to be more practical, but also to have a bioprotection role. It may, however, have an impact on the volume of the sludges, and especially if you don't control the temperature well and you have an early start of the fermentation. So control of the temperature below 10 Celsius is very important during the whole pre-fermentation stage. Move with experimentation B. We had here a very similar protocol, still Macabeo 2020 in France, seems still the same strain. Everything is very much similar. It is, however, a different must, and I will present you the data. We had 4 different inoculation modes in this experimentation B. In scenario number 5, the yeast was prepared without rehydration, so direct pitch, into the must at 10 Celsius. So that's DP10, direct pitch 10. The scenario number 6 was after settling without rehydration, so direct pitch, but after the temperature rose to 16 Celsius, so it's DP16. Scenario 7 was after settling with rehydration in cold water at 18 Celsius, and then we waited until the temperature rose to 16 Celsius to inoculate, so that's rehydration 16. And finally, the last one, number 8, is again with rehydration in warm water at 35 Celsius and an acclimatization phase before pitching in the must at 16 Celsius. So it's acclimatization 16, and this is still the recommendation that is done in order to inoculate a yeast to enologist. The average must analysis, here you have the data. Like I said, it's slightly different. We have a concentration of sugar or 243 grams per liter. Again, here we adjusted the YAN from 100 to 180 ppm through the addition of DAP at 1/3 of the fermentation, and we adjusted the turbidity. So here we have the result of the microbial population during the fermentative phase. So the first graph shows the population at 2/3 of the alcoholic fermentation. And we can see that the total yeast population were very similar between the four scenarios and very high. So in the order of the 8th to the power. And when we look specifically at the population here on the right, you can see that 95% of the population in all cases were Saccharomyces cerevisiae, so quite a high percentage. And when we look into the detail of the strains, we can see that here we can see some difference. In the case of the direct pitch, so these first two bars, we see a very high and good implantation of 100% of the HDA 54 in the total Saccharomyces cerevisiae population. In the second scenario, where we had rehydration and acclimatization, we can see that this drops to 40% and 10%. And in this case, we identified 4 different Saccharomyces cerevisiae strains. So here, something that I didn't mention earlier is that between the scenario direct pitching at 10 Celsius and the time that it took to warm up the water, to let to let the must warm up to 16 Celsius, and the time to prepare the yeast for rehydration and acclimatization, there was an hour to an hour and a half difference between the scenarios versus the direct pitch in 10 Celsius. And most probably in that time was a very good time for the strong yeast population that was present to develop. And that could explain this difference. So here we have the kinetics of these 4 scenarios, and we can see that actually the 2 scenarios, direct pitch, were indeed faster to ferment. We start here at day 10, and we're ahead of the 2 scenarios with rehydration and acclimatization, and ended at 17 days, when the 2 other scenarios took 4 more days to finish. So we see significant difference on the kinetics. On the analytical parameters here, we also see some differences. The main one I wanna discuss here is the volatile acidity that you can see in the scenarios where we had direct pitch and we had really good implantation of the HDA 54. We see lower volatile acidity than the rehydration and acclimatization, where we had 4 different native strains overall identified that could explain a higher level of volatile acidity. So some significant differences. On the dosage of the ethyl ester, here again, same way of presenting than earlier, we do not see some significant difference between the scenarios. However, we do see some trends for the ethyl butanoate and ethyl hexanoate, where we have higher production of these ethyl esters for the direct pitch scenarios, where the yeast implanted better, there was a better production of the ethyl esters. For the acetate esters, that is again a key marker of this strain, we see some significant difference for the isoamyl acetate. Especially, there is a higher production for the scenarios, the direct pitch, where the yeast implanted better. Now we move directly to a descriptive tasting, again with 18 different variables, 7 professional taster, and an ANOVA statistic analysis. And here we present the result for the amylic perception in the nose and in the mouth. And you can see that the 4 different scenarios belong to different groups. So between the scenario DP10 belong to group A, for example, and rehydration or acclimatization B and C, and B, C, and C, they are judged as different by the panel. And same thing here, scenario 5 and 6 belong to different groups than rehydration. So we see significant differences for the amylic perception. Really, in this case study, specifically, where we analyze the mode of preparation of the yeast, we show some significant difference. I do want to say that it is not always the case that we do this. We have our easy-to-use certification on yeast, and we do not always see a difference between rehydration and direct pitching positively toward direct pitching. But in this case, we did. So we saw some differences on the fermentation performances. On the implantation of the active dry yeast, the direct pitch had a better implantation versus rehydration and acclimatization, which directly had an impact on the fermentation kinetics. We saw also some significant difference on the base analytic parameters, such as the volatile acidity, and on the aromatic molecules, especially the isoamyl acetate, and this directly translated into a significant difference into the organoleptic profile. So on the final conclusion on this project, Safeno HDA54 can be inoculated in bioprotection mode on grape. I think this was showed in study A. In that case, the control of the temperature during the pre-fermentation phase is very important at less than 10 Celsius to avoid the start of the fermentation and settling issues. We showed also that this yeast can help inhibit indigenous yeast development, and the best implantation rate was when the yeast was inoculated very early on before pressing, but that inoculating after pressing was best for a fast alcoholic fermentation start. In all cases, a good implantation in the must was really related to the particular organoleptic profile of this strain, a high production of isoamyl acetate. Also, that the easy-to-use certified Safeno HDA54 yeast can be directly pitched in pre-fermentation stages. We showed that in study A, and in post-settling in experimentation B, without any implantation and fermentation issues. And in this case specifically, we saw that when inoculated with prior rehydration and acclimatization, some implantation issues can be encountered, sometimes leading to deviant profile. Overall, this study shows that for certain yeasts, such as HDA54, the easy-to-use technology combined with early inoculation timing can guarantee the achievement of the targeted type of wine. So thank you very much. Again, I apologize I'm not able to be with you in person, but I will be here to take questions remotely if you have any. And if it comes later, here is my email address. Feel free to contact me later and come by our booth to ask questions later. Thank you so very much.